Hello guys, good morning. Sorry I was running just a little bit behind schedule. Babysitters didn't get here in time. Um, I'm going to put the test off until Wednesday. This morning, I'm just going to go over chapter 16, the rest of the chapters that are in the book. I'm just going to pre-record those for you guys. But the test is going to cover the last 4 chapters that we did. So those quizzes are going to go up probably in a few minutes right after your class. And so I'm going to go over chapter 16, which deals with DNA. Even though y'all going to have quizzes on those last few chapters, I'm not going to give you an exam on them. I am going to post a 200 question study guide. That's what I've been really working on for you guys to review. Hopefully you guys can see me. Yeah, y'all can see, I can see the screen. Let me block it out. I'm going to post a review so that we're going to review next week. That's mostly what we're going to be doing. So you guys can meet, ask questions, but I'm going to post a review so y'all can get started. The test, because the power went out last night and everything over this way, I was doing some stuff and everything just shut down. I'm going to just go over chapter 16, so I didn't get a chance to post everything like I wanted to because power was going out and everything was crazy last night. So let me open up chapter 16 and we're going to get started, and that'll be the last chapter that, because it's a big chapter, that's going to be the last chapter that I go over completely with you guys. Your test is on Wednesday. We will still meet, but what we're going to do is I'm just going to tell you about the review and everything. So it's going to be a short meet, but it's about the review and what's going to happen for the final exam. Your final exam is going to be in 2 parts. It is 100 questions, so you're going to have a part 1 and a part 2. So it's going to be 100 questions, and those questions are going to come from the 200 question study guide. So it's going to be in a PDF format. You just need to answer the questions and go through and do the study guide. So once you do the study guide, that's what you're going to have. Okay, let me pull up the chapter 16. So that'll be the last big chapter we go over. All the other chapters are short, and I'm just going to pre-record those. So give me just a second. Okay, so the last major chapter and the thing that, you know, we see a lot of is really dealing with DNA. And DNA is an indispensable tool that we can use when it comes to dealing with criminology and dealing with different things. And so it is very extensive. Um, so that's one of the things we're going to look at. Why is DNA so important? Why is that? So this is one of the main things that really make or break cases. I mean, this day and age, there's a lot of people who have gotten out of jail because of DNA, and that was wrongfully accused. And then when they went back and tested DNA, people have actually gotten out of jail. And so you see that a lot. So let's get started with that. So a human cell contains two types of DNA. You got my nuclear and mitochondria. We will first focus on nuclear DNA. Now, nuclear DNA is very large molecule. They are linked in a series of repeating units. Those repeating units are called nucleotides. Now, a nucleotide is composed of sugar, a phosphorus containing group, and a nitrogen containing molecule called a base. There are four types of bases associated with the DNA structure. That's adenine, guanine, cytosine, and thymine. Now, I talk about these a lot in biochem, so Annalise, get ready. Go ahead and start learning those. You will see those again. So that's just four. It's actually, they make up what we call amino acids. And so that is just 4 of 21. Now, how do they be linked to form the DNA strand? S designate the sugar component that is on there, which is joined to the phosphate groups to form the backbone of DNA. Projecting from the backbone are those 4 amino acids, those 4 bases, adenine, guanine, thymine, and cytosine. Now, the DNA molecule is composed of 2 strands wrapped around in the other. They form what we call a double helix. The bases on each strand are properly aligned in a manner known as complementary base pairing. So as a result, adenine pairs with thymine and guanine pairs with cytosine. Now, let me get to this. So now this is how the double helix look. Notice how the bases G and C pair with each other, as do bases A and T. This is the only arrangement in which the 2 DNA strands can align with each other in the double helix. It has to line up in a certain kind of way. Now, when it comes to DNA, it directs the proteins. That's what it does. So, which are made by combining the amino acids. Now, when it comes to the amino acids in a protein, it determines the shape and the function of the protein. So the amino acids have to sequence in such a way. And so those 4 that I was talking about, that was the basis, those are amino acids. All right, so now each group of the 3 nucleotides in the DNA sequence codes for a particular amino acid. So GAC codes for amino acid glutamine, where CTG codes for alanine. So each 3 groups of those nucleotides bases, that's what they do. They code a certain way. If the nucleotide is changed, and we have that, GAG becomes GTG. The wrong amino acid is placed within the protein. Um, in this case, glucamine is replaced with valine. As a result, the protein may not function correctly, and that's what happens a lot of times. And this is the basis for many types of diseases and different types of health issues. Now, here you see a string of amino acids composed of one protein chain of hemoglobin. And this is what you see here. So you see normal hemoglobin, and then you see sickle cell hemoglobin. And you see what happened with sickle cell. Glutamate here is replaced with valine right there. And that's what happened. That makes the sickle cell long and odd shape. So that's what happens. Now, DNA duplicates itself prior to cell division. Now, DNA replication begins with the unwinding of DNA strands of the double helix. We call that denaturing and unfolding. So denaturing is one of the things we say. Each strand is now exposed to a collection of free nucleotides that is used to recreate the double helix letter by letter using base pairing. So that's what happens. Many enzymes and proteins such as DNA polymerases are involved in unwinding the DNA, keeping the DNA strands apart, and assembling the new DNA strands with the proper order, A with T and G with C. So that's what happens there. Now, once the strands of the DNA molecule are separated, the two new strands are assembled. Now, let me go back. Okay, so that's what happened. And they have polymerase chain reaction, or PCR, is a technique for replicating small quantities of DNA or broken pieces of DNA found at a crime scene outside a living cell. So PCR is used a lot. I'm even starting to talk about this in biochem. So polymerase chains reaction is the outgrowth of knowledge gained from an understanding of how DNA strands naturally replicate within a cell. For the forensic scientist, PCR offer a distinct advantage in that it can amplify minute quantities of DNA many times, a lot of times. First, the DNA is heated and separated. Second, the primers, now that's the short strands of DNA that used to target specific regions for the replication of the DNA. They are added in, and then it is hybridized with the strands. And then these are different primers that you can use. Now, DNA, it does repeat, and they have these tandem repeats. And portions of the DNA molecule contain sequences of bases that are repeated numerous times, known as tandem repeats. Tandem repeats seem to act as filler spaces between the codon regions of the DNA. So that's what happens there. Now, to a forensic scientist, these tandem repeats offer means of distinguishing one individual from another due DNA typing. What is important to understand for a particular tandem is there is a tremendous amount and variation in the number of repeat units that they have. Now, this right here is just a DNA segment consisting of a series of repeating DNA units. In this illustration, the 15 base core can repeat itself hundreds of times. And that's what they usually be talking about. Now, now, to the mid 1990s, tandem repeats were known as restriction fragment leak polymorphisms, or RFLP. And that was mostly the focus of anybody that was working in DNA at the time. And there are link differences associated with that. So that was one of the bases for the first type of DNA procedures. Typically, you have a core sequence 15 to 35 bases in length, and it repeats itself thousands of times. Now, the key to understanding DNA lies in the knowledge that there's a lot of possibilities. Number of times these repeat itself. So it's just a lot of possibilities that can happen. So that is what you really should look at and see with that. PCR technology just cannot be applied to RFLP DNA typing. With RFLP, the strands are really too long and to offer a number of bases. Hold on just a second, you guys. Okay, now PCR is best used for when DNA strands and that is just short. So basically, no more than in a couple of hundred strands. Now with that, the short tandem repeats. Oops. Oh, it's clicking too fast. Let's go back. Okay. All right. Sorry about that. My thing trying to get away with me. Hold on. And it just ran away for some reason. All right. Let me go back. I wasn't trying to go that far. Okay. So what happens is you got these short tandem repeats. So it's the latest method of DNA typing. Uh, it's most successful and is used in DNA profiling. There are locations on the chromosomes that contain short tandem repeats, and they repeat within the DNA molecule itself. Now, STR normally consists of repeating sequences 3 to 7 bases in length. And the STRs are much less susceptible to degradation. Also, because they are short, they are ideal candidates for PCR. So that's why a lot of people like using this over that. And because of that, is more so associated with crime scene evidence. The STR is a 2-band pattern that they use. So the mixtures with that, it's just simpler. STR pattern arrives from 2 individuals. It'll have 4 peaks. So that's one of the advantages of it. Is just that it is just simpler and easier to use and everything. Now, um, there are different types that we like to use. One of them being THO1, and it contains the repeating sequence AATG, and then 7 of them have used to identify the human genome. And these variants contain 5 to 11 repeat units. And so when you look at DNA, it's just a lot of things. You're looking at the Human Genome Project, all of that. People have been able to really do this. 2 such variants contain 6 repeat units, and then you got other ones that contain 8 units. During a forensic examination, the THO1 is extracted, and then it's amplified by PCR, and then it's separated by electrophoresis. And this is what you see here. So what you see here is this repeat unit. Now, this one is designated at THO168. Now, by examining the distance that STR has migrated on the electrophoretic plate, one can determine the number of ATTG repeats in the STR. And so everybody usually have 2. You have one that is inherited from each parent. So that's what you have there. And so you have one from mom, one from dad. So this one right here, 6-8 is found in about 3%. Of the population. What makes STR so attractive to forensic scientists is that you can find a lot of them in the genes. You can find a lot of them in human genes. Now, the more STRs you can characterize, the smaller will be the percentage of the population from a particular combination of STRs can emanate. Now, what about this? You can do multiplexing, and you can use technology of PCR. You can extract and amplify a combinations of different combinations of STRs. So that's the power that this really has. So like I said, this did a lot of things with making or breaking different cases, freeing people. A lot of people really been using this technology. Now, this is a triplet system containing 3 loci. And so what they did here, FGA, VWA, and then this is an indicator, a match between question and the standard reference strands right here. Now, a high degree, when you talk about product rule, you can have a high degree of discrimination and even individualization by analyzing STRs. So you can do the multiplexing that way. Now, because they hold on just a second. All right. Now, because these STRs, they can operate independent of each other, the probability of biological evidence is really determined by the frequency of the occurrence in a population. The greater number of STRs um, that you characterize, the smaller the frequency occurrence. Now, with that, now, what about the testing? Now, they have standardized this. Now, in the crime labs, they have 13 STRs for entry into the national database. That's CODIS. In 2017, it would increase to 20. Now, a high degree of discrimination and all of this multiplexing can determine the product of their frequencies. So a lot of this have already been entered into CODIS, and they are doing even more because we have technically mapped the human genome. You can do a lot more. With STR, you just need as little as 125 pictograms of DNA. This is about 18 DNA containing cells. You do not need much at all. Now, this is a STR profile for 15 loci right here. And then what about other things? Now, DNA can tell you the sex. That's the aminogenin gene, which is actually the gene for tooth pulp. It is shorter by 6 bases in the X chromosome than in the Y chromosome. When you amplify that by PCR and separate it, males who have X and Y shows 2 bands. Females that have the 2X just have 1 band. So that's how you can determine the sex determination. A male will have 2, female will only have 1 because they have 2 X chromosomes. Now, why do they like to do this? Especially when it comes to sexual assault, rape, the STA pattern arising from 3 males will only have 3 bands. So they can also type the STR located on the Y or the male chromosome. And so that Y STRs, they are useful for multiple males that is involved in sexual assaults. All right, change. Now, another tool available in the arsenal of DNA is the ability to type the STRs located on the Y chromosome, which is male specific. And there are more than 400 different YSTR markers that have been identified. The YSTR proved useful with multiple males in sexual assault. A YSTR analysis will have 1 band of peak rather than the conventional STR would derive from the 2 peaks. The YSTR is therefore less complicated and in appearance and interpretation. So they do use that a lot when it comes to sexual assault cases. Another type of DNA is mitochondrial DNA. Second, I had a student email me something. I needed to answer. All right. Okay, so mitochondrial DNA, mtDNA, is located outside of the cell's nucleus. Mitochondria comes from the mother. Mitochondrial structure found in all our cells, they use to provide energy that our bodies need to function. So we get that part of our DNA from our mothers. Mitochondrial DNA typing does not approach STR analysis in its discrimination power. And so that is used for hair. So that's what they use the mitochondrial DNA for. So they use that for like hair samples and things like that. Now, forensic analysis of this is more rigorous. It's time consuming when compared to nuclear DNA. So if you got people that, say, for example, my brothers and sisters that come from the same maternal lineage, it'll be hard to distinguish it from the DNA that way. Oh, my screen went out. Just a second. Okay. All right. So with that, two regions of mtDNA, HV1 and HV2, have been found to be highly variable, and a procedure known as sequencing is used to determine the order of those pairs. Now, every cell in the body got tons of mitochondria. It's tons of it, and this just shows you this right here. It can contain numerous copies of the DNA. The DNA in this case is shaped in form of a loop, and there's distinctive differences in individuals that make up the DNA as well. Now, one of the things that we're going to talk about is CODIS. Now, what they do is in CODIS, anytime you have committed a crime or anything like that, this is put into CODIS. And CODIS is just one of those things that they use the DNA and they put it in there to look at convicted sex offenders, other criminals. It's Combined DNA Index System. It's the software developed by the FBI. It contains local, state, national databases of DNA profiles, unsolved crime evidence, missing persons. Just from profile from unsolved cases right now is about 710 of them. It's a lot more. It's close to probably 1 million now. U.S. crime labs have standardized 13 STRs for entry into the national database, and I said in 2017 it went up to 20. So there are 20 of them in there. Now, when you get DNA, there are different sources of it. You got skin cell, hair, bone. So these are the different sources of DNA when you collect it. You can get it from the blood, semen, saliva, hair, skin, nail. I mean, you can get it from there. DNA is a powerful creator of physical evidence at crime scene. You can get it out of bottles, cans, glasses, cigarettes, bite marks, envelopes. Doesn't matter. You can get it from there. When you collect evidence from victims, this is where you can get it from. You can get it from dry blood. You can get it from clothing. Now, dry blood, you remove it from a surface by using a sterilized cotton swab moistened with distilled water. And then that is air dry before placing it in a swab box, then in a manila envelope. So that's normally what they do with that. Standard reference DNA samples got to be collected with a buccal swab. And so you swab the mouth or the cheek. So that's what the standard reference DNA samples have to be taken from there. Now, before you collect biological evidence, you got to photograph everything, record sketches. You got to do all of that. You got to wear nitrile gloves. Now, in the lab, that's what I only let my students use. I don't use latex gloves are there, but in the lab I like it because you have to wear powder free, because the powder can get on there and contaminate it. So that's why I like wearing those myself, and have my students wear them in the lab. Um, Now, usually when you do that, you have to double glove. And what they like to do is you have the other glove on to protect you. And then at that top glove, you always change out. Now, what else do you wear? Um, Face mask, lab coat, eye protection, shoe coverers, coveralls. You have to wear all of this stuff. So just like we are out of school dealing with the coronavirus right now, the same thing apply. You got to have personal protective equipment on. And so you got to wear all of that stuff. This is just so important when you're handling biological samples. Now, uh, when you're doing this, you got to collect your blood evidence. You got to collect all of this stuff. This stuff is very important. Now, clothing from the victim and the suspect with blood evidence, you got to collect all of that. You got to photograph stuff. You got to do all of that. The packaging of the biological evidence in a plastic or airtight container must be avoided, because the moisture can build up and you can get mold, mildew, and that can destroy the evidence. So you've got to make sure you pack it tight and airtight so no moisture or anything get into it. Um, Contamination, double layer gloves, face masks, all of that stuff, changing the outer glove, collect a substrate control for possible subsequent lab examination. So you gotta minimize contamination. That is very important. And the last thing, you pick up small items of evidence, just as cigarettes, butts, stamps, all of that. You got to pick them up with forceps. You got to dispose of them so that after that, and you always package each item in its own ventilated container. So when you're dealing with DNA, it is very important that you collect it in with that. So that's what you have to do with that. Now, like I told you guys before, chapter 16 says out of all the chapters, chapter 16 was the longest one with 66 slides. Chapter 18 and 20 is very short ones, like 30 slides a piece or smaller. Everything else I am going to pre-record. So the rest of the chapters are going to be recorded and posted on my website, on my YouTube channel. So they will be there. What I want you guys to do, the test is technically supposed to be today, but because the power and stuff went out, I'm just going to delay it until Wednesday. I'm going to go ahead and get it together and post it. So it is going to go up. I am going to give you guys some quizzes to do. So the test will be due later on this week. So I'm gonna give you the quizzes for the rest of the chapter. So those gonna be loaded. You work on the quizzes. The test will be loaded up on Wednesday. Like I said, what I am doing the rest of this time is basically reviewing things that is going to be on the test. I am going to make you guys a 200 question study guide. That's what you need to study with. So I'm getting ready to work on that and your quizzes. So I'm going to post the rest of your quizzes today and all of that stuff so that you guys can get ready for your test and your final. So your test is going to be due toward the end of this week, and then we're reviewing. So we're going to still meet for classes, but we're just going to be reviewing, going over questions and stuff from your first 4 tests and what's going to be on your final exam. So that'll be questions for you guys to ask then. That is all that I have for now. Like I said, I'm going to pre-record the other chapters and I'm just going to post those. But for the next 3 class meets, what we're going to do is review what's going to be important on the test, what topics, the main things I'm going to pick up, the question. So I'm going to get all of that together for you so you guys can be reviewing for your test. If you got any questions or anything, please let me know. Drop me an email. I'll get back to you as soon as I can. That's all that I have for today. Let me go ahead and try to convert this and get this up for you guys and get everything ready. I will talk to you guys maybe shortly on Wednesday for your test. So just make sure you log back on and I'll talk about the test and start reviewing for the final. You guys have a good one. I will talk to you guys later.